good afternoon. Am I audible to all of you? Yes.
Yes, ma'am, you're audible.
Am I audible?
Yes, ma'am, you're audible, ma'am.
So, respected principal of the institute, yes. Respected principal of the institute, Dr. B.T. Zadhav, sir. Vice principal, Dr. V.Y. Deshpande, sir. The chairperson of the Research Advisory and Development Committee, Dr. Mujawar, sir. All my colleagues, students of BSc and MSc. So, today's program,
I think that Madam was offline.
बाहेर पडी.
Lalkar madam, I think that madam left the meeting.
मीटिंग मध्ये आहेत ते, त्यांचं माईकचं माईक
अनम्यूट झाला की काय? का म्यूट झाला?
माईक दिसत नाही. व्हिडिओ आहे.
hello.
हा मॅडम.
Yes, repeat.
Hello.
Yes, ma'am, you're audible now.
Yes, yes. There was a problem in the network, so I was disconnected for some time. So now today's program that is being scheduled for the TYBAC and the MSc students of life sciences, as per the guidance of the director of the institute, Dr. B.T. Zadhav, sir, a very enthusiastic, workaholic personality, always trying to give the best to the students to keep them in pace with the modern world. I'm thankful to the director, YCIS, Dr. B.T. Zadhav, sir, Dr. Mujab, the research development, for allowing me to shoulder the responsibility of organizing today's national webinar on recent trends of research in life sciences. We have with us two very eminent personalities in the field of microbiology. The HOD of microbiology of Shivaji University, Kolhapur, a renowned person in the a figure which is well known in research, and Dr. Amarja Zoshi, scientist C at the NCCS Pune. I'm feeling very proud to state that Dr. Amarja Zoshi is the alumni of the Department of Microbiology. And both these persons, the resource persons, will guide the students to show you the horizons and depth of the research in the field of life sciences. I welcome both the resource persons who relentlessly accepted our invitation, and I'm sure that you will get the best of the knowledge of research that is going on in life sciences today itself. I know that we are facing the situation of COVID. We have been locked down since last 1 or 1.5 year, but definitely these days also will pass away, and we will be again back to our work, back to the academics, back to the research, where these research guidelines will help the coming budding students, I'm sure. So with these words, I welcome the resource persons again, and I hand over the charge to Ulalkar madam. Yes, Ulalkar madam.
Oh, yes. Thank you, ma'am. Now I request Honorable Dr. B.T. Zadhav, sir, Director, Yashwantrao Sawan, Institute of Science, Autonomous Satara, to deliver the inaugural speech. Over to you, sir.
Thank you, madam. On behalf of Yeshwantrao Institute of Science, Satara, and Rate Institute of Research and Development, once again I welcome all the participant delegates, research students, my colleagues, and honorable resource person, Dr. Kailas Sonone sir, as well as Amarja Zoshima from Pune. Dear friend, this is a very good opportunity to all of you because Research and Development Committee has organized very wonderful activity for life science students and the researcher. Dear friends, our college is always trying to give the fruitful things to the students and the researcher by organizing such wonderful activities uh, in during this even though this pandemic situation. We know that life science is a very big subject. It includes the botany, zoology, microbiology, biotechnology, and so many allied subjects related to microbiology and other subjects also. This is very vast subjects, and nowadays pharmacy is also included, medicinal sector is also included in life science, so that you have a very good opportunity to do the research activities under life sciences. And so many applications of the biotechnology are there. Recently, microbiology is focusing on the industrial microbiology. Botany is focusing on the plant protection and seed technology. Zoology has so many uh, branches as well as so many research wings. So, by such activity, our research and development committee of this institute uh, is going to organize this wonderful activity. And dear friend, we have the PG at 4 departments. We have the PG at biotechnology, at microbiology, as well as at the botany and zoology. So my opinion regarding this subject is that you should link your research activities to the recent trends. What are the recent trends? Whatever I read from last few days is that recent trends are focusing on the uh, medicinal sector as well as the pharmaceutical sector as well as the as well as the industrial sector, and everyone wants that your subject should be linked to the industry. Even botany, if some people are thinking that how we can link botany to the industry, so my suggestion is that the farmer should be linked via botany to the industry. Zoologists should link to the industry via some sericulture projects as well as some uh, innovative projects, that is vermiculture may be there, vermicomposting may be there, and that industrial microbiology should be linked to the industry. The subject should be linked to the industry. So when we link to the industry in future, it is necessary to learn the subjects in a deep manner, and we have to focus on the research activities which are helpful to the society. So many people are saying that computer people, electronic people, chemistry people, they are doing their research for the society. But I think that life science is also helpful for life science research is also helpful to the society. And whatever what may be the problem is that suppose that there is a guide and he had done his PhD 10 years before or 20 years before, and what happened? He is giving his extension, his topic extension to the student, and the student is not interested into that topic who is studied by the guide and supervisor long back ago, and the problem creates here, and so that student is not taking any interest into the research activity. So that is not my opinion about guide, but many times what happen, uh, teachers, old teachers as well as the brilliant people who have published their articles in the so many reputed journal, they are extending their research problem through the student, and they are taking the help for their extension of the research student. So researcher, even though research supervisor and guide have to focus on the new topics, then and then that topic may be helpful for the society, and society will accept your research activities. And if society is accepted your research activity, definitely industry come to with you, and industry people will discuss with you. And my another suggestion is that, or it is my opinion, you have to interact with the industrial people. And we know that in every industry there is a small unit that is called like the R&D unit of that industry, and that R&D unit of that industry going to focus on the recent trends, whatever the recent trend they knows, and we are lagging behind the industry. So industry and the educational institute must run parallelly. Whatever I am saying is that industry and institute run parallelly, not to be in the sense of walking. Walking is very dangerous for the research activities. Some people are walk slowly, some people walk slowly. In our fuzzy language, some people are walking step by step. Walking is not helpful for the industrial people. So we have to run by taking the hand of the industry. This is helpful for any subject. This is not only for the life sciences, but also for the all subjects. And my third thing, my suggestion is that you should publish the paper into the reputed journal. But before publishing your research article, the industrial people are filing their patents, and they are getting the benefit of the patent. They are focusing on the RPR. But we are our research is only for the publication of the 2 article, and guide is suggesting that you have to file the thesis to the university, and university will award the degree to you. My suggestion is that your research activity may not be up to the up to the degree, but you have to extend that activity for the patent, for the society. So these are the things in the recent trends, for recent trends in every subject, in every research. So once again, I congratulate to the Mujawar sir and Kanshe madam for organizing wonderful activity. And Sonamne sir is always helping and motivating for the department. And even though for the so many departments of the university, he is leading the position of HOD. And microbiology people are doing well. They have the deep thinking, they have the deep knowledge of the subject. As compared to other subjects, microbiology had a lot of opportunity. And under the able guidance of Dr. Sonamne sir, we will also do some research activity in our institute. Thank you, Sonamne sir, for supporting us. And once again, I welcome all the participants and the students. And I declare that the one day research activity and the workshop is inaugurated. Thank you. Thank you very much. Thank you, sir.
Okay.
Thank you, sir. Thank you.
Thank you so much, sir. And I thanks to all of you behalf of the organizing committee. Now we'll move towards the session first. So I request Ms. Manisha Shinde ma'am to introduce our today's chief guest, Honorable Professor Dr. K.D. Sonone sir, NIH Postdoctoral Fellow USA, Professor and Head of the Department of Microbiology, Shivaji University, Kolhapur, and Honorable Dr. Amarja Zoshima ma'am, Scientist, CNCMR, NCCS, Pune, India. Over to you, ma'am.
Thank you, ma'am. Good afternoon, one and all present here. Today, we are honored to have eminent resource person for one-day national webinar on recent trends of research in life sciences. And I'm very pleased to have a chance to introduce them. Today, first eminent resource person is Honorable Professor Dr. K.D. Sonone sir. He is head of the Department of Microbiology in Shivaji University, Kolhapur. He has great experience in field of research. He was awarded with Indo-French Project Fellowship, CSR Senior Research Fellowship, and Postdoc Research Fellowship by National Institute of Health in USA. He has 76 international and 4 national research publications and 3 international publications of book chapters. He also contributed in development of biological databases. He has submitted 11 gene sequences of microbial species to GenBank. He had delivered talks in national and international conferences. So with this brief introduction, I heartily welcome you, sir. Our second eminent resource person is Honorable Dr. Amarja Zoshima. She is currently working as a scientist C at National Center for Microbial Resources, that is NCMR NCCS Pune. She has research experience of 12 years in field of microbial taxonomy, systematics, and ecology of microorganisms, particularly isolated from extreme conditions like Lonar Lake. She has 14 research publications in reputed peer-reviewed journals. She also guides students for master dissertation projects. She was awarded with Research Associate and Senior Research Fellowship by CSIR. And I feel very privileged and proud to share that she is alumni of our college. With this brief introduction, I welcome you, ma'am. Actually, today it is an honor and privilege for us to have both of you with us. Student, you also feel enriched with knowledge after completion of this webinar. So let's have a pleasure of listening to both of them. Thank you so much.
Thank you, ma'am. So now I request Honorable Professor Dr. K.D. Sonavane, sir, to continue with this talk. Over to you, sir.
Okay, so first of all, good afternoon, everyone. And actually, I am very much thankful for this opportunity. Particularly, I thank here Honorable Principal Dr. B.T. Jadhav, sir, for organizing as well as supporting this lecture series. I am also thankful to Dr. Mrs. Kanse Madam as well as Dr. Mujawar, sir, for organizing this wonderful lectures for all MSc students. And first of all organizing committee of this particular lecture series, thank you very much for this wonderful opportunity to interact myself with all students, particularly UG as well as PG students. So students, as we know, the topic of this particular seminar series or lectures that I have been given by Madam, particularly Kanse Madam, is that is recent trends of research in life sciences. So as our honorable principal sir has said, life sciences research is a very vast area, and actually it would not be, I mean, one cannot cover most of the things related to life sciences research area. There are n number of research areas right now available, right now present. Various researchers are doing research in these areas. So so I will be covering only few research areas, and so that most of the research subjects, like particularly botany, zoology, microbiology, biotechnology, biochemistry, they will get some kind of benefit from this particular research areas. All right, so let me share my PPT. All right, can you see the PPT?
Yes, sir.
Okay, all right, thank you very much again for this opportunity. So I will be talking on recent trends of research in life sciences. Okay, so first of all, let us see what is meant by research. Okay, since there are many UG students, are the, I guess, as well as PG students who will be getting degree of master's degree after a few months, or maybe who have got already degree in this particular their particular discipline. So let us see what is this, what is meant by research. So research is a commonly one can call as refers to search and for knowledge or for new knowledge. One can also say that scientific and systematic search for pertinent information on a specific topic. And at the end, I would only say that it is, some people consider research as a movement from the known to the unknown. And finally, it is actually voyage of discovery. Okay, so what makes people to undertake research? So there are certain possible things, motives for doing research, maybe either one or more of the following. Maybe desire to get a research degree along with its consequential benefits. Some people may think that after doing research, I may get some degree, PhD degree, all right, and that would be beneficial for my future career. On the other hand, some people may think that desire to face the challenge in solving the unsolved problems, that is, concern over practical problems, initiates research. So that is more important if I consider. Similarly, another important aspect, that desire to get intellectual joy of doing some creative work. This is another important aspect, right? One should have a desire to get some intellectual joy, or to learn new thing, or to create some new knowledge by doing some creative work, or to desire to be a service to the society. So as we know, we are getting degree, we are getting jobs, right? Maybe we get salary, each and everything. But what if we should also give something back to the society, whatever society has given to us? So that is, one should have a desire to be a service or to the society, and desire to get respectability. Okay, so I consider 2, 3, and 4 are very important aspects that why people make research. All right, now there are many more factors. Similarly, like directives given by government, that government says that every faculty should have PhD degree, every faculty should do research. All the employment conditions, some of the employer may ask that you should do research, all right, you should publish research papers. Then curiosity about new things. There are some faculty members, some scientists, they have curiosity in their mind that they said, let us do something new in the particular thing. Desire to understand casual relationships, all right, or social thinking of awakening, and to like, maybe as well as motivate. So motivation is essential part in this case, I would consider. All right, Okay, so there are, as I said earlier, there are n number of research areas are available in life sciences. So it is not right time, or it is this 1 hour will not help us or help even me to cover or cover all this point related to. So I will just brief out some of the research areas, that major research areas, active research areas that are most of the research scientists, faculty members are doing active research from universities as well as research areas. So first of all, their focus is on enzymology, that identification, isolation, characterization of different enzymes from different sources. So that sources could be botanical plant sources, animal sources, or microbial sources. And those should have some industrial importance. That means industrially useful enzyme. This is one of the very active area of research that one can say right now. Then most of those scientists, they work on biofertilizers, biopesticides, right? Application, the degradation policies, bioremediation is another very good research area. Then few researchers are working in this particular area, particularly our department of biochemistry, department of biotechnology. They are well known in doing research in this bioremediation. Then phytochemical analysis and their uses as an antimicrobial agent. This is very important area right now, because as another research area, that is antibiotic resistance. So I'll be talking more on this particular antibiotic resistance in this particular talk. So as we know, this antibiotic resistance is one of the major public health threat nowadays. It is being said that after 5 years we won't be having a single drug, single antibiotic to tackle, to control, or to kill pathogenic microorganisms. So it is a very essential, it is the right time to discover a new drug that we can consider as an antimicrobial agent, right? And that can be isolated from maybe different plants, the secondary metabolites from different animals, or secondary metabolites from different microbial sources, different microorganisms. Okay, so these phytochemicals would play a very important role in the present situation. Then antibiotic resistance, this is a research area now. It is a hot topic right now, I would say, and I'm glad that Department of Microbiology, Shivaji University Kolhapur, have started working on this antibiotic resistance way back from 2012. All right, and we have got very good results on this in this particular area. And most of the life sciences researchers, they can contribute equally in this particular area. It is not like that only microbiology staff or scientists they can contribute, but no, plant scientists, animal scientists, that means botanists, zoologists, microbiologists, or chemists, they can equally share their thoughts in this particular research area. So I would consider this is a very important area. Then neurodegenerative diseases like particularly Alzheimer disease, which also I am going to talk in this particular talk, because we have started working on this Alzheimer disease way back in 2016 after my joining at Shivaji University Kolhapur as a new research area at Shivaji University Kolhapur, and we have really got very good results in this particular disease called Alzheimer disease. What are the causes? What are the mechanistic approaches that we are basically focusing about the Alzheimer disease? So I will talk on this particular disease. This is another important area where most of the researchers are doing research. World-class institutes, they are doing their best to understand specific reasons about the Alzheimer disease. Then nanobiotechnology and their medicinal applications is another important application, budding area or hot topic, I would say. And I would like to inform you that Dr. Mrs. Mane Madam, we can call it Kanse Madam, she is doing very good work in this particular area. She has produced one PhD student, very good PhD student, and that candidate, I think Nilofer Nadaf, has got very good research papers in this area. So synthesis of nanoparticles from different sources, again, plant materials, animal sources, microbial sources, and analysis of those particular things, nanoparticles, and their medicinal application that one can talk about. Then what are the methods are essential? Which method that one can target? So some of the advanced methods of bioinformatics that I will be talking in this particular talk, that what is bioinformatics and how these methods can be useful to understand some of the deadly diseases, their molecular mechanisms, right, in detail at the molecular level. And finally, whatever research area that we do, basically our aim is to design a new drug molecule, drug discovery, right? And how these techniques, how these areas, and what is the progress in particular drug designing, the drug discovery, and how much is the time that one can get to discover, design and discover a new drug molecule. Basically, our final aim, final goal is new drug molecule, so that one can control growth of pathogenic microorganisms or some of the deadly diseases. Okay, so everyone's has got final goal: design a new molecule, lead molecule. As we know, we are going through this COVID-19 pandemic, and some of you might have vaccinated, and few companies are trying to get some new drug molecules in order to cover or tackle this particular problem. So this drug discovery and designing is a very important area that I will also talk few things about these things. So these are the advanced areas that maybe today we are going to cover. And last one, taxonomic studies. So these taxonomic studies done by different scientists, maybe botanists, zoologists, microbiologists, they all do, and it is very, very essential area. Because of this area only one can try to understand new species, new organisms. One can understand their phylogeny and evolutionary aspects. One can try to understand. All right. So this is another research area. Although I'm not going to talk more about this thing, only these highlighted areas by yellow color that I will be talking more in this today's talk. Okay, so let us begin with Alzheimer's disease. It is one of the neurodegenerative disease. Basically, one can call it as a memory loss in this particular thing. So this Alzheimer's disease is a complex progressive neurodegenerative disorder. It is common cause of more than 60% to 70% dementia among all the affections. About 36 million people worldwide, they have been affected by this particular disease. It is estimated that by 2050, 1 in every 85 people will be living with AD or with Alzheimer's disease. So pathological consequence of this AD is due to aggregation of amyloid beta peptide. It is a small peptide which contains either 40 or 42 amino acids, and deposition of this toxic assemblies into as the extracellular plaque near our brain cells. Okay, so deposition of these plaques leads to the formation of neurofibrillary irreversible damage to the neurons. So available drugs also interfere in normal functioning of the brain. And so particularly right now, gut microorganisms, they have been known or they have been discovered, they can play a very important role in this particular AD. So this some part also we will discuss in this particular today's talk. So there are some basic thing that I will explain, and then we'll move towards which method that one can apply and what are the results that are currently available. So as we know that there are a few major hypotheses of this Alzheimer's disease, particularly cholinergic hypothesis. As we all know, acetylcholine is one of the neurotransmitter which has to be transported from one cell to the other cell in order to transmit or transfer messages. So if something goes wrong, then one cannot have some proper movement or proper transportation of this acetylcholine. So that leads to the cholinergic hypothesis. There is a tau hypothesis. Tau is a protein linked with the our microtubules. If that tau protein gets more phosphorylated, that means addition of phosphate group, then that tau things get disturbed. Our microtubules get disturbed, and this would lead to the formation of some neurofibrillary tangles, and that can cause interference in between two nerve cells. That is another hypothesis, tau hypothesis. And third one, that is the most important one, that is called the amyloid hypothesis. In this amyloid hypothesis, what happens? There is a amyloid beta protein. It is the one small protein which contains, there are two types of proteins, amyloid 40 and amyloid 42. So these two proteins, if they try to come together, all right, there is many major secretion of this amyloid beta proteins, and then they can try to form kind of a structure, and that meshwork kind of structure, or we can call it as a blocks, that can try to block the nerve cells through passing through messages from one nerve cell to the other nerve cells. So this is one of the major aspects that right now many researchers are working on this amyloid hypothesis, and in Ma'am. Hello, can you hear me?
Yes, sir.
Okay. Thank you. All right. So this is a particular that we were talking about this, how amyloid precursor protein called as a APP.
Sir,
Right?
share your screen.
Okay, share. Okay,
Screen is
screen
not
sharing has
visible.
gone. Okay. I think screen sharing has gone. Okay. All right. Now, is it visible?
Yes, sir. Visible.
Okay, sir. Okay, thank you. So this amyloid precursor protein, after cleavage with certain enzymes, then we get a small peptides like amyloid beta peptide. So this single amyloid beta peptide then try to clump together, that there would be formation of oligomers. Many more such amyloid beta peptides can be formed, and then they can try to come together. They can try to form oligomers, and then there would be formation of fibrils, and that fibrils would lead to the formation of plaques. All right, and this is a particular thing, plaques formation, and then one can say that the person is suffering from Alzheimer disease. So here is our healthy cell, healthy nerve cells, that one nerve cell can communicate properly with the another nerve cell. But if person is suffering from Alzheimer disease, then this is the plaque, right? A kind of a meshwork kind of a type of structure formed by the oligoformation of fibrillar formation of different amyloid beta peptides. Okay, and so that this is, we can call it the diseased nerve site. Protein are widely transmembrane protein having a molecular weight to 130 kd. So amyloids are proteolytically derived from specific enzymes called as the beta secretase and gamma secretase on amyloid precursor protein, and which would lead to the getting of small A beta peptides. So this deposition of this A beta peptides leads to the formation of plaques. Okay, so here we can see that. So this is the amyloid plaque, right? One single A beta peptide, if they try to come together, there is a formation of amyloid plaques or beta pleated sheets of the structure. As we know, most of the students, they know that there are two types of secondary structures, alpha helix and beta sheets. Alpha helix is the helical rod-like structure, and beta sheets are fat, flat and elongated structures. So this is our membrane, and this is the APP, amyloid precursor protein. If by the action of beta secretase, we get APP beta. And then again, after gamma secretase, this A beta 40 and A beta 42, two monomers that one can get. If these two monomers try to merge together, try to come together, and they form amyloid beta oligomers. And this amyloid, if there are many more such oligomers they synthesize, then there would be formation of amyloid plaque, which leads to the neuronal dysfunction or causing kind of a disease. And this is just the primary structure of these proteins, that A beta 42, which contains 42 amino acids, and A beta 40, which contains 40 amino acids. So both the proteins, they try to form amyloid plaques. So there are certain, but now what is the treatment? So some treatments are available right now, but not that much effective. But amyloid beta aggregation inhibitors are there. So as we can see, hybrid peptide inhibitors are there, or polyphenolic compounds. Maybe polyphenolic means from plant species that one can get. Okay, so plant botanist can try to, contribute here by getting or by discovering new phytochemicals, so that would try to form or inhibit this particular formation of amyloid plaque. Nanoparticles are also being used as one of the aggregation inhibitors, right? Maybe our efforts are going on into discover, synthesize new nanoparticles to avoid any aggregation inhibitors. Then there are certain degrading enzymes. Particularly, we are working on some enzymes that, right, this aminopeptidase, gelatinase, catechin, all the enzymes that we have worked, and we have tried to discover that how this enzyme can degrade these amyloid beta peptides in detail at the molecular level. So there are certain peptides from microorganisms as well, right? So our efforts are also going on to identify or discover new small peptides from different microorganisms, and that small peptides can be useful or could be useful as a one of the inhibitory factors for this aggregation of amyloid beta peptide. Okay, there are small molecules like curcumin, ibuprofen. These are some of the molecules are also scientists have studied. Antibodies work is going on, and peptide inhibitors are also going on. So there are, as we can see in this particular area, many researchers, many faculty members, they can contribute in this particular research area, so that most of the researchers, particularly botanists, zoologists, microbiologists, biotechnologists, they can try to contribute in this particular field, particularly to understand more in details about the Alzheimer's disease. Okay, so let us move further. And there are role of bacteria as well. So recently it has been observed that one specific bacteria called P. gingivilis, a non-motile gram-negative rod-shaped anaerobic bacteria, which has been identified from the mouth, right? So which can leads to the secretion of more amyloid beta peptide. So there is a very good important role of microorganisms in this particular aspect in AD. So most of the researchers are also doing some research work in this particular thing. And not only in detail, I will just explain in shortly that when harmful, particularly E. coli gram-negative bacteria secretes some lipopolysaccharide, and that lipopolysaccharide can activate the some pathways that we will not go into the details about these pathways, and cause intestinal wall and epithelial inflammation. So that could also lead to the formation of Alzheimer's disease to the specific individuals. And similarly, there is the gut microbiota secreted polysaccharides and other toxins which can cause neuroinflammation and Alzheimer's disease development. So that means definitely there is these are the recent discovery. Recently, it has been identified that role of microorganisms in amyloid beta peptide. Why someone gets Alzheimer's, why other doesn't, right? It actually depends on gut microbiota as well as some of the microorganisms that or more formation synthesis of peptides. Okay, so I will not go into aspects about this one. So now let us see how to do research, or some of the research aspects of this particular areas. So from this slide is just for the student purpose, that as we know there are 20 different amino acids that make up the proteins, and one amino acid is linked to another by a peptide bond. Since we are talking about the proteins as a drug, okay, so this is our primary structure and the linear sequence of amino acids. From primary structure, we try to form secondary structures. Alpha helix is rod-like, helical rod-like structure, and then beta sheet-like structure. If such secondary structures try to form tertiary structure or three-dimensional structure, which is actually functional form, and if we have more than one polypeptide chains, then there would be formation of quaternary type of structure. All right. So, and this is called as the dynamic behavior of the proteins enzyme that one can study by using this particular technique, some of the tools. So this is just a simple example, hemoglobin, right? Here we can see heme group along with brown color one, and blue one is oxygen. So as soon as there are certain conformation changes take place, there will be two changes in conformation, oxyconformation and deoxyconformation. So such conformational studies that one can do in detail at the molecular level using some of the techniques, advanced techniques which are available, and same technique one can also use to solve this particular Alzheimer's disease problem. Okay, now whatever drug that we discover, so that drug basically, if you go by traditional drug discovery methods, this particular one can start with target identification, target validation, lead identification, candidate optimization. Then one can go for preclinical studies, clinical studies, and whatever data is being generated using these processes, that we can call as a data mine. That is another area of research. Okay, and so if we complete all these particular steps, and afterwards any pharmaceutical company gets their drug into the market after spending nearly 15 to 16 years of time, and that is very longer period, and to do that pharma company has to spend more than $800 million, right, to get single drug into the market after spending 15 to 16 years of time. But if you combine these methods along with today's computer-aided drug designing methods, that application of computers, that may be bioinformatics methods, chemoinformatics methods, metabolomics, genomics, proteomics, these are all advanced areas, advanced research areas. If we combine all those methods along with the traditional drug discovery, one can discover any single drug within just 5 to 6 years of time. So that imagine, so see the difference, right? The 15 to 16 years time, after spending $800 million, what your drug that we discover that would finally that would create some side effects. So FDA, that is Food and Drug Administration, says that, yeah, let us ban on this particular drug. So imagine what would be the condition of that particular pharma company after spending $800 million and 15 to 16 years of time. So in order to avoid this, what pharma companies have started, they first of all sit in front of computers, use some of the sophisticated softwares, maybe freely available or commercial or paid softwares, and they design a drug, right? The way we design a building, right? First of all, we map, we draw the structure, we design a building structure, and we can think about how much of material is required to construct this whole building, how much period it would require, it would take. Similar to that, one can also design a new drug molecule by considering our receptor, right? And receptors as discovered by some of the biologists, maybe botanists, zoologists, microbiologists, or biotechnologists. And then one can easily go for clinical studies and clinical studies. So one can discover a new drug molecule within a 5 to 6 years of time. This is the advantage of some of the, if we use computer-aided drug discovery. Okay, so let us discover what are the methods that one can see before going to the actual. We have discussed about Alzheimer's disease, and we have discussed about drug designing, drug discovery, what time it takes. Now let us see which methods are there or advanced methods in life sciences research to do or to design a new drug molecules. So there is one area called as a bioinformatics. It is a combination of biology and informatics sciences. So biology means it is nothing but botany, zoology, microbiology, biotechnology, all right, all that biology knowledge. If we combine that knowledge with little bit knowledge of informatics sciences, maybe some physics, some chemistry, computer science mostly, all right, that would give us a new discipline called bioinformatics. It is the application of information technology applied to the management and analysis of biological data with the help of computers. So the key to biotechnology discoveries is locked in the genome of various organisms, and bioinformatics holds the key to unlock this data for the next generation of innovations. So let me repeat these two sentences. The key to biotechnology discoveries is locked in the genome of information technology applied to the management and to understand biology in a better way. All right. So why it is essential? Why one should learn bioinformatics? One can predict 3D structures of our industrial proteins, enzymes. One can predict function of newly discovered proteins. One can predict gene function, establishment of gene libraries. One can produce ability to use genome sequences to identify their function, protein-protein interactions, modifications, and their function. And finally, elucidate the function of a molecule based on the structures. And one can also use some of the hardcore methods of bioinformatics called molecular modeling methods, molecular dynamic simulations, conformational search using quantum chemical methods, molecular docking to understand protein ligand interactions. So we'll talk something about this molecular docking method in this particular talk today. So what are the major areas where students can seek job opportunities, like molecular biology databases, sequence comparison and analysis, structural bioinformatics, and drug discovery. So these are the biological data that we deal with, particularly proteins, nucleic acids, carbohydrates, and lipid molecules. And if someone, any scientist, any faculty member, if he or she wants to set up bioinformatics laboratory, right, then they need a simple things. What are things? High-speed computers they require. They require internet connection. They require some freely available as well as commercial softwares, UPS to avoid power fluctuation, and air conditioners to maintain room temperature for this particular high-speed computers. So if we have only this much things, one can set up our bioinformatics research laboratory. All right. So what is the hub or major area, major institute who have started particular work of this particular bioinformatics created by National Center for Biotechnology Information, created in 1988 as a part of National Library of Medicine at the NIH, right? Where I got an opportunity to work as a postdoctoral research fellow during the 2003 to 2006. So their goals were to establish public databases, do research in computational biology, develop software tools for the sequence analysis, and disseminate biomedical information. Okay, and this is a particular website that most of the students, particularly UG as well as PG students, they would require to collect information or to collect references in order to start their research project. Because I know that every MSc students, but doesn't matter whether it is botany, zoology, microbiology, so every research student has to perform research project at the end of the year, their MSc second year. So now question is that from where they can download research articles, research papers, from where they can get some more information about the what is happening recently, or what are the latest happenings in the research areas. So this is the entrance search engine called as the life sciences search engine, where most of the students, they can get a lot of information from this particular, they can download whole research papers from this PubMed Central. They can identify which scientist working on in which area, how many research papers he or she has published. All that information one can get through this search. NCBI entrance search engine, Google and Yahoo for the commercial search engine. These are the commercial search engine, but this is the search engine made only for the life sciences students. All right, and now there are different biological databases. Now what is database? Databases are nothing but an effective electronic filing cabinets where a convenient and efficient method of storing large amount of data, a large amount of information. So there are some types of databases. I will not go into details of each and everything, but only thing is that for student purpose, this is how primary structure of proteins can be isolated. One can identify sequence, and that sequence one can deposit into the primary protein sequence databases. So here this is called as the FASTA sequence format, which starts with a greater than number, GI number, name of the protein lysozyme, each source, and this is the primary structure of protein, starting with methionine. Then we have lysine, amino acid, phenylalanine, valine, isoleucine, right? These are different kind of 20 amino acids, and so student must know first letter codes of each and every amino acids and their physicochemical properties. Intention in showing this particular slide just to keep in mind that student must remember all the 20 amino acids along with their sequence, structure, function, and physicochemical properties if they want to do good research in future. All right, so that is a new area called as the proteomics, that if we have a whole set of proteins, and that whole set of proteins we can call as the proteome of that organism, and the study of that whole proteome using experimental and computational technique is called proteomics. Okay, similarly, if there is a total set of genes present in that particular organism, that would be called as the genome of that organism, and study of that whole genome would be using experimental and computational technique is called as the genomics. So as we know, our human genome is known right now, right? The human genome contains 30,000 genes, 3 billion chemical base pairs, which could code for 1.5 million proteins. So there is a huge task in front of us to identify location, mapping of that genes, structure, function of each and every gene, each and every proteins of only humans. Forget about other microorganisms like E. coli genome, yeast genome, rice genome, wheat genome. All genomes have been known now. And now there is a huge task in front of all the students, all research faculty members, to try to understand structure, function, role of each and every protein, each and every gene belonging to all these particular organisms. All right, so this is a sequence analysis one can perform, sequence comparison by taking multiple sequences. One can try to find out conserved regions, active site of the enzymes, metal ion binding sites, cofactor binding sites, all right, vitamin binding sites. All these binding sites one can try to find out using sequence analysis, one of the important technique, important area. And now if we know the 3D structure of the any proteins, any enzymes, one can try to find out a function of that enzyme. So most of the pharmaceutical companies nowadays, they are going behind understanding drug and receptor interactions. So drug, it could be anything, any drug, any small molecule, and receptor could be any protein, any enzyme. All right, so that protein enzyme interactions one can try to find out by using some of the advanced techniques. Okay, so once we determine 3D structure, one can try to find out active site of the particular enzyme, and this, as we see, these few residues try to come together and they can form active site of the enzyme, where substrate or inhibitor going to bind to that particular enzyme. Okay, and then one can compare a real model versus our model by using superimposing techniques. So here two structures have been superimposed, and one can see that they are exactly identical or common in nature. Not only that, one can also design now question is that how to design a drug molecule. So there are certain commercial softwares through which one can design and discover a new drug molecule. Right? By if you know the molecular structure of the drug, one can get three-dimensional structure. One can generate three-dimensional structure of this particular drug, and one can visualize in different models like ball and stick model, wire model, tube model, or space field model. And finally, this is our drug. Now we have a drug is ready. We have designed a new drug by considering its molecular structure, which has got three dimensions. Three dimensions means having XYZ Cartesian coordinates. All right. So such drug, now we are going to take it further to interact, to identify its interactions with the receptor. All right. And the way we study enzyme kinetics inside the test tube, right? So similarly, one can also study such interactions using bioinformatics method called as the molecular dynamic simulation method. So I will not go into the details of this particular method, but only thing is that here we can see protein or enzyme surrounded by water molecules. So it could be a buffer solution or our water molecules. And if we add any our buffer or any inhibitor, any enzyme, one can try to find out how this enzyme interact with the particular drug molecule. Okay, so there is another technique called as the molecular docking technique, through which one can try to find out how one protein interacts with another protein. Let us say this is one protein or one enzyme or receptor, and this is our protein as a drug. So how this protein interact with this particular receptor, and that we can call as a protein-protein docking. This is one of the very important technique right now. Most of the pharmaceutical companies they are using to design, discover new drug molecules. On the other hand, if we have a small drug, take an example of paracetamol or any other drug, right? Donepezil in case of Alzheimer disease. And if we have a receptor, right? So how this drug interact with this receptor, that one can try to find out, and one can optimize or one can try to energy minimize, and one can get most stable structure of this particular compound. Okay. So, and this is how enzyme that one can see. This is our enzyme and this is the drug. So this drug has inhibited the pathogenicity of this particular enzyme, that we can call as the enzyme drug receptor interactions or protein drug interactions. And this is how this active site of the enzyme and some of the residues from this particular enzyme protein can how tightly bind with the particular drug molecule. And the most of the stick model that we can see, these are all amino acids, and this is the one enzyme through which we can block the harmful activity of this enzyme by using this drug molecule. And this is the just space field model that you might have seen such figures in various textbooks, that how the enzyme can be blocked by the drug. So this is a space field model or surface structure, and this is a brown color is our drug molecule. And such studies can also be done to identify or to design a new drug within a short time. So that's why the area called as a computer-aided drug design is very essential area, important area, where one can study or use most of the advanced methods and get design a drug molecule, and so that one can design, synthesize or discover that drug within a short period, maybe 5 to 6 years of time. So there is another technique called as the virtual screening technique for drug discovery. That means what happens here, if we have a, let us say, one enzyme and we have 1 lakh drugs. So that means one has to, if we follow traditional drug discovery methods, one has to perform 1 lakh time of experiments, that one enzyme and 1 lakh drugs. But before that, if we sit in front of computers and use this technique called as the virtual screening technique, one can try to find out of this 1 lakh drug, which is the drug can be beneficial or can fit properly into the active site of this enzyme. This technique is called as a virtual screening technique. This technique is also being used very widely in most of the pharmaceutical companies. All right. So this is how one can determine 3D structures of any proteins, any enzymes by using most of the bioinformatics methods, and one can try to find out, compare your discovered drug molecule along with already experimentally available drug or drug which is already available in the market and which we have discovered a new one. So this is where one can try to find out which drug can interact properly or suitably. Okay, so let me now let us turn to our Alzheimer topic, right? So just I have discussed about which method that one can use to understand this Alzheimer disease problem, right? What is the exact cause and what kind of solutions that one can get after using these methods. Okay, that was my intention in telling first of all. So we have discussed first of all some of the introductory aspects of the Alzheimer disease, some methods of the Alzheimer disease that we have discussed, and now let us see which techniques that one can go. All right, so this is the one area that we have used. We have used that is called as the structural analysis of a membrane-bound HEC protein, that is a human endothelin converting enzyme, which is the one enzyme which can degrade amyloid beta peptide. And this is the overall enzyme, and this enzyme can cleave amyloid beta peptide, which is one of the causative agent of Alzheimer's disease. So what we have done, we have predicted three-dimensional model, three-dimensional structure of this enzyme. All right, and we have to try to find out how this amyloid beta peptide, a causative agent of Alzheimer's disease, can enter inside the enzyme, and how this enzyme can cleave amyloid beta peptide as a therapeutic agent. All right, so this work has been published in Amino Acid Journal, a very good journal having high impact factor 3.9. And similar to that, we had published one research paper in Neuropeptide Journal. Right, Sagar Barge, one of PhD students, had done this particular work, and so this work has been published in Neuropeptides. So this Neuropeptide Journal, they try to publish, let us say, 1,000 research papers per year, and out of these 1,000 research papers, they select based on content or content of the particular paper top 25 hottest articles. And as we can see here, our paper highlighted as the #1 research paper, right? By considering only content of this particular research paper. That means if we try to maintain quality of the research paper, research work, try to use advanced methods, one can get very good research papers, and such research paper can be shortlisted as a top 25 hottest article, and that too, still this paper is #1 article among the top 25 hottest article. Okay, now in this particular Alzheimer's disease, there is a role of microorganism there, or microbiology role is there. So particularly this is for microbiology students, that how they can contribute in this particular field by considering knowledge of microbiology. So this is another study which we had published in Computers in Biology and Medicine. Again, this is a high impact factor journal. So what we have done, a simple thing, that homology modeling, molecular docking, and MD simulation studies to investigate a role of cystine protease. This is one enzyme from Xanthomonas campestris in the degradation of amyloid beta peptide, a causative agent of Alzheimer's disease. So here we have taken a sequence of microorganism, right? The first sequence that we can see, and second sequence from human Alzheimer's disease patient, right? Cathepsin D sequence from Alzheimer's disease. Now we have come system, and Alzheimer patient is system, but still they can possess some of the common thing, that is common active site enzymes. So what we had done, we had predicted three-dimensional structure of this cystine protease and tried to dock this amyloid beta peptide, brown color that we can see, and this is the cystine residues present in the enzyme can cleave amyloid beta peptide into smaller fragments. And if we get small fragment, such small fragment can be thrown out through the blood-brain barrier, through the circulatory system, and so that one can that patient can get relief from the Alzheimer's disease. So this paper has also been shortlisted as the top 25 hottest article, but featured as the number 12 first year, and then second year number 21. So by considering, since it was the first report that we have shown how microorganisms can play important role in the Alzheimer's disease. Similarly, there is another enzyme from microbial species called aminopeptidase. This also we have studied, and the same work again published in Amino Acid Journal, and the enzyme aminopeptidase from Streptomyces regensis MK, which we have isolated, extracted from this Shivaji University area, and this is the species which we have identified, and the gene sequence of this species had already been submitted to the GenBank, and from this microorganism we have identified or discovered this enzyme called aminopeptidase, and we have studied how this aminopeptidase can cleave amyloid beta peptide, one of the causative agent of Alzheimer's disease. Similar to that, there is another organism called Stigmatella aurantica. It is a myxobacterium known to produce some enzyme, right? So this enzyme called as a gelatinase. So this such enzyme, or AC, angiotensin converting enzyme. So these studies have also been done. Another to that, we have used published very important work in RS Advances Journal, that is the Royal Society of Chemistry, in search into the molecular interactions of amyloid beta peptide degrading enzyme, that is gelatinase, from the Enterococcus faecalis. So this Enterococcus faecalis has got ability to produce this enzyme gelatinase, and this gelatinase could cleave amyloid beta peptide, one of the positive agent of Alzheimer's disease. So that means microorganisms species can play very important role in this particular area, and this is a mechanism that we have studied, how this gelatinase can cleave amyloid beta peptide in combination with the zinc. So zinc is very much essential, as you might have heard about one government ad, that जिंकी गोलीलो, एक गव्हर्नमेंटची ऍडव्हर्टाईज आहे की जिंकी गोलो जो प्रेग्नेंट वुमेन किंवा त्यांच्यासाठी आहे की जर तुम्ही जर झिंकची गोळी घेतली तर होणारं हेल्दी बाळ आहे ते हेल्दी होईल, इट वुड नॉट सफर एनी काइंड ऑफ डिसीज. So this is the role of zinc that we have showed at molecular level, that what is the role of zinc and by this enzyme gelatinase in this particular Alzheimer's disease. Now another question of that oligomerization or aggregation of inhibition. So another study which we had published in ACS Omega journal. Okay, so this is a small peptide. As I had said earlier, the small peptides could also be useful to avoid aggregation of this amyloid beta peptide. So there is a arginine 7 amino 4 trifluoromethyl coumarin, RRAFC. This is a drug peptide with another one. Okay, so this insertion of RAFC into hydrophobic core is stabilized by amyloid protofibrils. So this is a small drug molecule which can intercalate in between this amyloid beta peptide. So such studies that we had done, and it has been published. Now, is there any treatment available right now? What is the recent treatment? So recently in March 15, 2021, an experimental drug appeared to slow cognitive declination, and that name of that particular drug is nothing but, yes, donanemab. So this is a recently discovered drug in 2021 itself, which has been shown to prevent amyloid beta peptide formation. Okay, all right. Now this much was the thing about related to Alzheimer's disease, that what is the exact cause, what is Alzheimer's disease, and how one can understand this role of this particular disease, and what are the different treatments are available. So similar to that, there is another area that which I am going to talk today, that is nothing but antibiotic resistance or drug discovery. So these antibiotics, as we know, there are streptomycin, gentamicin, kanamycin, different antibiotics are there. So these antibiotics, some of the pathogenic organisms, they have developed resistance, right? So how to overcome that resistance, how to identify that resistance, and how one can discover a new drug molecule. So this antibiotic resistance among various infectious diseases or infectious agents towards different types of antimicrobial drugs has emerged as a global cause of public health threat. Antibiotic resistance is the ability of microorganisms to withstand the resistance of that particular antibiotic. So the diverse role, newly developed resistance pattern among microbial species and decrease in the efficiency of treating common infections disease resulted into the development of high levels of multi-drug resistant strains that we can call as the MDR strains. So there are different enzymes are involved in this particular thing. So let us talk of some of the mechanisms which are known in this particular area called mechanism of antibiotic resistance. So there are a few mechanisms. Like first of all, let us say this is our bacterial cell, as you can see, circular cell, and this is a cell surface. So this one, the antibiotics cannot enter inside the cell because of the decreased permeability of the cell. This is one aspect. Second one, even although bacterial antibiotic could enter inside the bacterium, right? But there are certain target sites can be changed, right? That means receptors can change so that antibiotics cannot act properly. This is second one. And third one, there are certain enzymes which can deactivate the enzyme and deactivate the antibiotics by changing its structure. So the antibiotic cannot act on that particular pathogenic bacteria. And last one, that is, although if there is entry to that particular antibiotic, but that antibiotic directly goes out to the particular cell through the active transport of the cell through efflux pump. So these are the four mechanisms of antibiotic resistance. That means certain pathogenic microorganisms, they can develop resistance by any one of these particular way. Either it could be decreased permeability, that means those bacterial cell cannot allow or do not allow that particular antibiotic to enter inside the cell, or they can change their receptor, the receptor or enzymes in such a way that antibiotics cannot be recognized, right? Or they can secrete some enzymes. Bacterial cell can secrete some enzyme so that that structure of antibiotics can be changed, or they can throw out the enzyme, or sorry, that particular antibiotic as it is, right? So these are the some four mechanisms for the antibiotic, and that's why this antibiotic resistance becoming one of the important threat or very major public health threat to the health sciences. So that's why it is being said that after 5 years we won't be having a single drug molecule to treat this antibiotic resistance problem. So it is now high time to all the researchers, particularly botanists, zoologists, microbiologists, or chemists, to come together and discover new drug which we could consider as an antimicrobial agent. So there are 3 enzymes that they can particularly target bacterial cells, like amino glycoside phosphotransferase, amino glycoside acetyltransferase, and amino glycoside nucleotide transferase. So we have focused on this particular one aspect, amino glycoside phosphotransferase, and these are the drugs which we have tested, like gentamicin, kanamycin, amikacin, tobramycin, streptomycin, and neomycin against specific organisms, particularly like Mycobacterium tuberculosis, Acinetobacter baumannii, Enterococcus gallinareum, and then E. coli. Okay, so these studies had been published in again another high impact factor journal called Journal of Cellular Biochemistry, and the work done by Rishikesh, and he had also discovered a new drug molecule from the using virtual screening technique, and with this drug showing more, or it showed more activity against routinely available drug in the market. Okay, so this is how sequence analysis can be performed. These are the method which I had just now discovered, and then this is the enzyme or model that we have discovered, and the residue, blue color residues can be shown or can be highlighted as the active site residues from this particular enzyme from Acinetobacter baumannii. Okay, and these are the drugs which we have discovered using virtual screening technique. This is the latest technique, and this is the drug which we have discovered, which showed more activity or potent activity as compared to the streptomycin antibiotic. Okay, so this is the known drug, tirfostin, and this is the drug which we have discovered. So our drug showed more potency against known drugs available into the market, and which this drug has been tested against different organisms like Mycobacterium tuberculosis, E. coli, Acinetobacter baumannii, and Enterococcus gallinareum. Okay, this is how one can try to find out mechanism of this particular drug. And now here, this cartoon figure, one would see severity of this particular problem, right? The currently chemical antibiotics are being used as the sole therapeutic components to control various infectious diseases. But many pathogens have developed resistance against these therapeutic agents. Okay, so antibiotic resistance a big threat to the society. So it is necessary to discover new therapeutic agents to fight against these pathogenic entities. So here, doctors with molded needle, right? And now this bacteria, pathogenic bacteria with covered, or we can call it a shield. So doctors are confused how to kill or how to control growth of this particular bacteria. So what would be the solution? So this smaller bacterial called as antimicrobial peptides, or AMPs, are the solution because these are peptides, and peptides now being targeted as effective drug molecule against organic components. So one can also, so most of the microbiologists, they can try to find out small peptides, and such peptides cannot be identified only from microorganisms, but from plant species as well as from animal species also as well. And this could be used as an antimicrobial agent. So herein we have identified a new enzyme, right? Antibiotic resistance and inhibition mechanism of novel amino glycoside phosphotransferase, APH5U, from the Bacillus subtilis strain RK. This is a new type of enzyme that we have discovered from this identified species, which we have identified, that is Bacillus subtilis species. And from this species, this is the enzyme which can possess or which is responsible to cause antibiotic resistance, and we have named it as the APH5. This is the first time enzyme that we have discovered at Department of Microbiology, Shivaji University, Kolhapur, by using all the experimental as well as these structural studies. And we have used high sophisticated equipment like LCMS, LC-QTOF. These are techniques have been used to identify actual mechanism. Similarly, most of the botanists can target on collection of phytochemicals from the different plants, right? So collection of phytochemicals and study those phytochemicals, purify them, and try to find out which phytochemicals are there, and then one can try to find out of these phytochemicals which one are more potent, more effective against certain pathogenic bacteria. So such studies we had published way back in the journal called Journal of Ethnopharmacology, one of the very highly reputed journal way back in 2018. And here we had also done some animal studies. So these are the phytochemicals that we have identified, discovered from the phytochemical from the plant, right? This was plant called as the Litoris, Hymenocallis litoris. This is a plant, and from this plant, these chemicals, phytochemicals that we have discovered, all right. And such bioinformatics studies can be performed to find out how they can interact with the pathogenic microorganism enzymes secreted by pathogenic organism like S or S, as well as next one, Candida albicans, right? Here Candida albicans that we had taken. Okay, now this was the area where most of the biologists, zoologists, they can try to give their contribution in not only Alzheimer's disease, but in case of antibiotic resistance as well. But let us say, for example, taxonomist, if botanist, zoologist, or any microbial person, if he or she, they have got more amount of data, more amount of information about the particular microorganisms, then they can also try to develop databases, right? Let us remember, which we had seen, what is meant by database. Databases are nothing but an effective electronic filing cabinet where large amount of data can be stored at a time. So we have developed one database called Mangrove Influent Database, right? A database of all mangroves medicinal plants with their sequence information. And the study had been done by one of my students, Sambhaji Thakar, now doing postdoc in South Korea. So mangroves are of great importance, right, to many people who live along our tropical shorelines. So mangrove plants are a rich source of various components having medicinal values. And in recent years, biodiversity and conservation of mangroves have been globally recognized as one of the critical issues. Hence, it is necessary to study different mangroves in detail at the molecular level along the taxonomical relationships. So this is how data can be collected, has to be collected from different sources, from plant source, animal source, or bacterial sources, all right? And then one can develop database. So this is how the way we have developed here. So one can, by clicking on single line, one can get a lot of information about this mangroves, mangrove species, mangrove images, contact, the administration, right? And this is the database which we have discovered, which have developed about the database of mangrove plants with protein enzyme sequences. Okay, so likewise, if we have a lot of data, so all the botanists, zoologists, microbiologists, they can also go into this area and they can try to develop various databases. All right, so I will not go into the details. And there is lastly some hardcore area that I would like to talk very little, maybe within 5 to 2 minutes, that I would like to comment on this particular role of modified bases present in transfer RNAs. So as we know, transfer RNA is a very good, very important molecule, adapter molecule involved in protein biosynthesis. Now this protein biosynthesis, as we know, it is a common process present in different organisms like plant species, animal species, as well as microbial species. So this is the cloverleaf model of transfer RNA, and in this transfer RNA, there are modified nucleosides are present. So what are these modified nucleosides? For example, these 3 different domains of life like eukaryota, bacteria, and archaea, they have been known they have been shown a presence of this such modified nucleosides. So this is a small base, and if there is the addition of methyl group, that would be modification. At the base of addition of methyl group, right, or addition of hydrogen, addition of one functional group, addition of amino acid itself, like lysine, right, or addition of methyl thiol group, that would give to the role of modified nucleosides, and that would give a complex structure to the transfer RNA. And this transfer RNA can play a very important role during the protein biosynthesis process. So this work that we had published in Cell Biochemistry and Biophysics Journal about the tRNA folding and the role of this modified nucleosides, particularly guanosine, if it is there, and modification of that guanosine. So I will not go into the details of each and every aspect, but at the end, only I would like to highlight some new aspect which we had discovered at Department of Biochemistry or Department of Microbiology, Shivaji University, Kolhapur. So as we know, Watson and Crick had developed one model, DNA model, in 1962, right? And they had got they had shared Nobel Prize for that particular discovery. But Francis Crick, second author from this one, Watson and Crick, in 1966 proposed the Amoled hypothesis. So what was his hypothesis? That this is the ribosomes and this is the mRNA. All right. So one tRNA comes, recognition of codon anticodon takes place, and then that tRNA goes out. All right. But he did not show, that Francis Crick did not show how it can enter and how it can leave the process. That he did not show at that time. So model called as a footstep model, considering the Crick hypothesis. Right? Ovaling basis helps in easier dissociation of tRNA mRNA. So such studies were published. This is from E. coli, Integrative Biology journal that we have published. Another work from human sources published in PLOS One journal, very reputed journal. And another study here we have taken tRNA, mRNA, and rRNA. All right, and these are the hydrogen bonding sites that we have proposed. That is, initially it recognizes by 2 hydrogen bonds and then by single hydrogen bond. Okay, and this is the whole ribosomal machinery that I would say, like tRNA, amino acids, and then rRNA, that which we had taken in this particular thing. So what is our model? That footstep model we have proposed, that translocation of tRNA takes place from one side to the other side, that is A side to the P side, right? From one side to the other side, goes in a step-by-step manner. That is a footstep model. So this is what we have proposed by considering all these advanced methods, right? And this whole work done at Department of Microbiology as well as Department of Biochemistry, Shivaji University, Kolhapur only. And these students, all students are from our affiliated colleges only. Okay, and this is the part that we had published in Journal of Biomolecular Structure and Dynamics. And here one can see in the abstract itself, we have mentioned that we have developed a new footstep model, which would be the extension of the Francis Crick model developed by Francis Crick. Okay, similarly, there is another work that we had published in Gene Journal. Okay, now this role of modified nucleosides and antibiotic resistance, right? These are the combined. Now our efforts will be in such a way that how one can combine these two areas, that modified role of transfer RNAs and antibiotic resistance. Okay, so if we do good research, good quality research, recognition is, one can get recognition by good citations. So as we know, this cell biochemistry and biophysics research work done by one of my students, Bajrang. Now he did training from this IIT Mumbai. His work has been cited in prestigious journals, as we can see, like Cell 2018, it has been cited, having 38.66 impact factor. The Nature 42.7, Nature Communications Biology, Nature Scientific Reports, Molecular Theory. And similarly, another work published by Asmita, right, Cell Biochemistry and Biophysics in 2015. Her work has also been cited by Nature Reviews Microbiology, having 34.02 impact factor. Like if we do good research, highlight some specific point, right, maintain quality of the research, then one can get very good research recognition by citing and such authors have highlighted or short or cited our work. That is the beauty of work which we do at Department of Microbiology. And this is the facility available at Shivaji University, Kolhapur, Department of Biochemistry and Microbiology. So I will not go. So different life sciences related problems can be studied and analyzed. One can understand structure, function, and dynamic behavior of biomolecules, drugs, as well as some of the cellular process in detail at the molecular level, using advanced techniques. And effective drugs can be designed using these particular techniques, which we can see. Now, within a few seconds, I would like to give one information to all students, which as Madam said, that there would be UG students also. So kind of a program that we teach at Shivaji University, Kolhapur, Department of Microbiology, called interdisciplinary programs. So we run by MSc Biochemistry, Biotechnology, Microbiology, and Pharmaceutical Microbiology as a concept of horizontal mobility. And recently we have started new MSc course, Medical Information Management, international course in collaboration with Hochschule Hannover University, Germany. Under this course, at semester 4th, our students go to Germany, and German student would visit Shivaji University, Kolhapur to complete their semester 4th. So this is in a collaborative research MSc project, student exchange program between faculty and students, which we had started way back in 2019. And so students can also try to get admission for this particular courses, and as well we provide fellowship to the students as well. So these are the research collaborations. At state level, we have 7 collaborations with Pune University, with ICT Mumbai, with Dr. Babasaheb Ambedkar Marathwada University, Aurangabad, Solapur University, SRT University, Nanded, Dr. D.Y. Patil University, Kolhapur, and Bharati Vidyapith, Kolhapur. Then at national level, CSIR, IICB, Kolkata, Indian Institute of Science, Bangalore, and National Institute of Virology, Kerala unit. Recently, we had published one good research paper with this NIV Kerala unit. And at international level, we have Aalto University, Finland, where we have published one research paper. The University of Jahangirnagar, Dhaka, Bangladesh, and KwaZulu-Natal University, South Africa. Okay, so these are the research students who have contributed in this particular area, and right now there are 8 students are working. And I would like to acknowledge Department of Science and Technology, University Grants Commission, New Delhi, as well as Lady Tata Memorial Trust for providing fellowships, HOD's Department of Biochemistry, Biotechnology, and our Honorable Vice Chancellor, Professor D.T. Shirke sir, for providing all necessary facilities, as well as Honorable Pro Vice Chancellor, Professor T.S. Patil sir, and as well our ex-Vice Chancellor, Professor D.B. Shinde sir, Professor M.M. Salungi sir, as well as Professor N.J. Pawar sir, and all Computer Center team for providing good infrastructure facilities. So thank you very much, one and all, for your kind attention, and I will be happy to take few questions.
Thank you so much, sir, for very informative talk. And now the session is open for the participants. So if anyone have a question, then you may put it in the chat box.
I hope I have covered everything in time.
Corey from the zoology. Yes, sir, you can ask the question. Doke, sir, please unmute Mr. Sunil Kori, sir.
Unmute करू शकतात सगळेजण. Allow केलेला आहे.
Okay. First of all, thank you so much, sir, for such a nice session. In one of your slide, you have mentioned about the aminopeptidase enzyme.
Right.
So I have one question about specifically about aminopeptidase P enzyme, which cleaves the N-terminal amino acid from the protein when there is a penultimate amino acid is a proline.
Okay.
So my question is, from which source I have get the information about the aminopeptidase P enzyme, which acts on which number of polypeptides, which number of proteins? Because in a on a Google Scholar, there are number of papers are there, but there is no any summarized information regarding aminopeptidase P enzyme, which acts on number of polypeptides. Specifically, the peptides, neuropeptides. So I am looking for the neuropeptides on which the aminopeptidase P enzyme is act.
So it is okay. Go ahead.
So if you know any source about that, where I can get all the information regarding aminopeptidase P enzyme, which acts on the number of neuropeptides.
Okay. This question is regarding getting sources, right?
Yes,
sources
right.
of your interested enzyme. So maybe aminopeptidase P or any enzyme. So as I said in this particular slide, right, this Entrades, it is a NCBI Entrades website, right? If you go to NCBI, National Center for Biotechnology Information, go to this Entrades, and this Entrades, one can find this PubMed. Okay, sir, here, this is a PubMed, and if you put on this PubMed your name of the protein, name of the enzyme, one can get how many scientists have worked on this particular enzyme, how many research articles are there, right? All that kind of information one can get at this particular website. It is called as a bibliographic or literature database. And not only that, if that journal, that website journal is freely available, then one can download that whole research paper as well. So this is the one site that where one can get this information. And there is a Web of Science is there, and Scopus. Scopus, it is a database which covers most of the journals that one can also get a lot of information from these 3 particular databases. Like PubMed is the one, and then Scopus another one. And there are certain research papers that one can also try to find out at the Web of Science. I hope this answers your question.
Yes, sir. Yes, sir. Thank you so much.
Yes. Good.
So next question. Okay, so I thought there is no question for sir.
Okay, right. That was Sunil Koli sir has asked question there from chat box.
Yes, sir. Yes.
Yes. Okay, sir. Okay, good.
Okay. So now I request Professor Mr. B.S. Pawar sir to deliver the vote of thanks. Over to you, sir.
Thank you, ma'am. May I audible?
Yes, yes.
Yes, sir, you are audible.
Good afternoon, one and all gathered here. I would like to present vote of thanks. It is a matter of pride to me that I got a chance to thank our first resource person, Dr. K.D. Sonone sir, who honored this function with his presence as well informative thoughts. Sir, I can surely say that this session is very informative as well fruitful for not only student but also faculties who start research newly. Thank you, sir, for your valuable time.
So with this, we'll move towards the session second.
Okay. Okay, thank you very much. Now I can sign off.
Yes, okay, sir.
Thank you,
Thanks a lot.
sir.
Thank you.
Thank you very
madam.
much.
Okay, thank you.
Thank
Fine.
you.
Okay.
So with this, we'll move towards the session second. Now I request Honorable Dr. Amarja Zoshima to share her knowledge with our students. Over to you, ma'am. Honorable Dr. Amarja Zoshima.
Has she joined? Yes, Tejaswini madam.
Yeah, what, ma'am? I can't hear.
Has Amarja Zoshi madam joined? Because I
Yes,
had
yes, ma'am. Yes, ma'am. She joined the session.
Yes, she was there because I had made contacts with her and she is there.
Yes, yes.
She'll shortly join, I think. Yes. We'll just wait. Yes, she's joining.
Hello, ma'am.
Yes, yes. Yes, you are audible. Amarja, you are audible. Yes. But we just cannot see your screen. We can just view your desktop. Yes. 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 I can see your screen. Yes. Oh, no, not very properly, rather. It's on the lower tone. Your voice is on the lower tone, Amarja. Yes, no, not very clearly. Hello, Doke sir.
Yes, madam.
हा का हो त्यांच्याकडून आवाज येत नाही आपल्याला?
मॅम त्यांनी जोशी मॅम तुम्ही जॉईन विथ ऑडिओ केलंय का?
आहे ऑडिओ दिसतो त्यांचा.
खूप कमी येतोय आवाज अमरजा मॅडम.
मॅडम तुम्ही लॅपटॉपला जॉईन केले का? हॅलो
हॅलो सर, एक 2 मिनिटं मी कोणाला तरी विचारलं.
जोशी मॅम.
हा हा.
हा जोशी मॅम एकदा काय करा की तुम्ही रिजॉईन करा एकदा.
ओके ओके, बंद करून जॉईन करू का?
हा रिजॉईन करा.
दोन मिनिटं येऊन जा. दोन मिनिटं मॅडम.
नाही मॅडम कोर्स झाला करा. करा मॅडम.
मॅडम ऑलरेडी आपण लॅब घेण्याचे सुरू आहे. आपण यामध्ये वेगवेगळ्या नोट आपण चालू करू. आलं मॅडम. एक दोन मिनिटं थांब. जरा फक्त इथं काहीतरी गडबड झाली आहे आवाजाची.
हा मॅडम आता येतोय आवाज तुमचा.
5 मिनिटं फक्त. हा मॅडम येतोय का आता आवाज माझा?
Yes, ma'am. You are audible now.
ओके ओके. ओके ना?
Yes, ma'am.
क्लिअर.
डोके सर परत कमी झाला काय आवाज?
आवाज
समजा 111 112 होते. अरे वा. Hello.
Yes, ma'am.
Amarja. No, I'm speaking with Amarja.
Yes, ma'am. Okay.
Teju, you can just tell the students to wait.
Yes.
Because her lecture will also be very, very nice for the students.
Yes, ma'am.
Hello. डोके सर त्यांना जरा थोडंसं गाईड करा की.
हॅलो.
हॅलो. हा.
हॅलो जोशी मॅम. तुम्ही जर लॅपटॉपवर जॉईन असाल ना तर हेडफोन नाही वापरला तर चालेल मॅडम. आवाज येईल व्यवस्थित.
Hello. Students are requested to wait for 10 minutes. 5 to 10 minutes. We'll start the session very soon. Please don't left out the meeting.
Hello.
हॅलो जोशी मॅम.
आमचे डोके सर वगैरे तुला इन्स्ट्रक्शन्स देतात ते ऐक ना जरा. हा बघते येत नाहीये का बघ बघू नाही तर मी कन्व्हेयर होते तुला. बोल. हा 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 डोके सर.
हा
हा त्यांना तुमचा पण आवाज येत नाहीये मॅडमना.
म्हणजे आपण बोलतो ते पण आवाज जात नाही की त्यांना.
हा आपला पण आवाज जात नाहीये.
त्यांना याला लेफ्ट व्हायला सांगा मीटिंग मधून परत जॉईन विथ ऑडिओ करायला सांगा.
ओके.
आणि बेटर लॅपटॉप असेल तर आता छान चांगलं होईल व्यवस्थित. त्यांच्याकडे लॅपटॉप जर असेल तर काहीच प्रॉब्लेम येणार नाही. आणि पीसी जरी असेल तर फक्त हेडफोन त्यांना चेंज करायला सांगा. दुसरा हेडफोन घ्या.
हा हॅलो ती परत रिजॉईन होतीये बर का सर.
बरं ठीक आहे ना.
Participants, please don't leave because really you will be listening a very interesting talk, very interesting research experiences from one of our alumni. She has graduated from here from Department of Microbiology. She is a student and she is doing very well at NCCS Pune. So please don't leave.
हा जोशी मॅम आवाज येतोय. हॅलो तुम्ही अनम्यूट आहात बोला. जोशी मॅम आवाज येतोय. हॅलो. मॅम पीसी वरनं जर प्रॉब्लेम येत असेल तर तुमची जे पीपीटी आहे ती पीपीटी मेल करा, इथून प्रेझेंट करतील. तुम्ही फक्त मोबाईलवर जॉईन करून बोला.
हॅलो डोके सर, त्यांना तुमचा आवाज पण जात नाहीये. काय असेल तर मी निरोप देऊ शकते तिला.
ओके.
आम्ही म्हणजे काय म्हणताय तुम्ही? तिच्या पीसी आहे तिच्याकडे, पीसी वरून तू जॉईन होतीये, ती ऑफिस मध्ये आहे.
लॅपटॉप असेल तर लॅपटॉप पूर्ण करायला सांगा.
लॅपटॉप नाही ना, लॅपटॉप नाहीये त्याच्याकडे. ती पीसी वरती तिचं काम चालू आहे, तिथूनच ती जॉईन झाली आहे.
एक काम करा, तुमच्याकडे पीपीटी घ्या, त्यांची पीपीटी तुम्ही शेअर करा आणि त्यांना फक्त मोबाईलवर बोला म्हणा.
बर, बघते मी, बघते ती शेअर करते का.
मोबाईलवर जॉईन करून बोला म्हणा.
हा हॅलो. हा बर, असं करू शकतेस का तू? पीपीटी शेअर करतेस का? हे जेवून घेतोय झाले असं जेवण जास्त येईल
जोशी मॅम ओके ओके तुम्ही मोबाईलवरून बोला चालेल.
हा हा सर ऐकू येते
आता येते मॅडम. ओके ओके
ठीक आहे. हा मी मोबाईल वरनं बोलते सर.
ओके ओके
हॅलो
हा येते मॅडम.
कॅन वी स्टार्ट सर?
हा सर
हा ठीक आहे चालेल चालेल. हा ओके सर. या ऐकू येते सर आता व्यवस्थित?
Yes, ma'am. Yes, you are audible now.
हो अगदी व्यवस्थित ऐकू येत आहे अमर्जा.
ओके सॉरी मॅडम.
No, it's okay. It is a technical issue that it can be raised for anyone.
Okay.
You can continue. Okay.
Okay, madam. Thank you. Thank you so much.
Okay.
First of all, good afternoon, everyone. And today I invite all of you for this national webinar on recent trends of research in life sciences. Before starting my talk, I would like to thank Dr. Kanse. We are calling her Mane Madam. So for her kind invitation for this today's talk. When I talked with ma'am about this topic, she told me to discuss recent trends and mainly different opportunities which students after MSc the students have in this exciting field. And when we discuss with each other, I was so happy to that madam gave me such an excellent topic for discussing with you, all of you, because after MSc, the main question is what to do next. Many of us don't have a particular plan of doing specific things ahead. So if we get some guidance about what to do and how to approach such things, then definitely it will be somewhat easier for all of you in to choose your career or to choose your path in future. So I will start with my lecture. Okay, so first point here is what is research? Research is nothing but a creative and systematic work undertaken to increase stock of knowledge. And here the words creative and systematic are very, very important, as both these things are very essential while undertaking any type of research. And this involves collection, organization, and analysis of this information to increase the understanding of topic or issue. So here, during performing any type of research, it is very important to collect the data, to organize it in a systematic way, and analyze the data to interpret some important conclusion. Okay, so this is what research is. So to perform any good quality of research, I think one has to have three qualities. I will say it as 3 P's, as passion, patience, and proactiveness. So what is passion? Passion is interest in performing various experiments, learn new things. So every one of all of us are passionate about different things in our life. But for research, you should be passionate about learning new things, and various experimentation like that. Why I say patience, a long process of about 5 to 6 years one should be patient enough to handle which occur during this period. And proactiveness. One should be very proactive during this process for taking up new challenges, learn new techniques, and collaborate with new people, and so on. So these 3 P's are very important to perform good quality research. Okay, so you will ask, what should we do once we complete our MSc? So there are two ways of going to it. As one can go as a technician in an industry or in government organization. So here you can join directly as a technician in an industry, or there are a lot of positions in government organization as well. Or you can join on one project which are going on in the government institutions. Second way is you can clear state eligibility tests or net eligibility tests examinations. If you are really interested in teaching, it's a very hard job, and if you are passionate about this, then you can definitely choose this career. I think for today's talk, this is very important thing. That is, one can join as a PhD student in national institutions throughout India by clearing the examinations for NET JRF. So one can go and join in such institutions by clearing Junior Research Fellow examination conducted by National Testing Authority. So this is very important to make a specific timeline to join all these things, because there are age limits for every position in government organization, like 28 years for technician and project assistant, and so on. So you should be very careful while choosing your path after MSc. So now, if we are as talking about research and PhD, then PhD, where should one do and why? As I already mentioned, one should do the PhD in national institutions throughout India. And this is because national institutions have complete infrastructure and facilities to perform research. They have expert staff for discussing your issues during your PhD tenure. You may get opportunities to interact with researchers in and outside India. And this is very important to make your collaboration very nicely with all these researchers, and which will be definitely useful in your future. Then here you can expose to newer technologies through various national, international conferences as well as workshops. So if you would like to do PhD, please go to national institutions, clear the examinations, and get admission. Okay, so here, if one would like to do PhD, it is first thing, it's a very long process. And so when we are doing our PhD, we always say that the long form of PhD is not Doctor of Philosophy, but it is Patience Highly Developed, because it's really a long process. It's a very hectic, and hard work and sincerity is very essential for completing this thing. So I divided these things into pre-PhD, during PhD, and post-PhD. So this is, every phase is very important, as in pre-PhD, you have to do rigorous referencing for deciding and selecting a topic which is not explored earlier. So topic selection is very important. Then you have to write a proposal, present a proposal, proposal and get approval from university for doing PhD. Then during PhD, this is a very important phase, as here you have to actually you have to do all the important things during this phase, as you have to do planning of work, performing various experiments, learn new techniques. Then here you have to observe and analyze and interpret your results. Very important in case of by doing your doctoral work, because it should be very perfectly interpreted. Then only you can get next objective. You can go to the next objective. So here, manuscript writing and publication. This is a very important stage of your PhD, because here you must write a manuscript, publish it in very authentic and very good impact factor journal, and this will automatically authenticate your work and increase your confidence. Then here, next is you have to organize and present papers at various national international conferences to develop collaborations with international experts. And the last stage is compilation of data, thesis writing, and submitting it for evaluation. And here PhD Viva to get PhD award. So these all are very, it looks like very simple steps, but this is not a simple, and it really needs a very hard work, dedication, and sincerity and patience. So when you enter in this stage, you are a simple MSc student, but when you come out of this phase, you really become a very thoughtful, skillful, and mature person. So I think PhD is very important stage of everyone's life. And if you are really passionate, please go for it and do the research through your PhD. Then once you complete your PhD, you can join as, this is the post PhD phase, where you can join as assistant professor at university level, or you can join as a scientist at industry or government institution. And one can do postdoctoral research in various organizations in India or abroad. So this is all about PhD and research work, how to start research and finish it in the during PhD, but it never finish. It will again continue in your future work as well. Okay, so to join a PhD or join as a PhD student, there are very renowned examinations which are conducted in India, that are CSR UGC NET examination, DBT biotechnology eligibility test, and GET. So these are main examinations. There are n number of examinations for entering in this area, but here I will explain these important first. This is a national level test. It is organized by NTA, that is National Testing Authority India, and it is conducted to determine eligibility for JRF. That is, here if you clear UGC JRF examination, you can join as a CSR JRF, and you can get fellowship and do your PhD in any Indian in any national institution throughout India. Or if you clear UGC, CSR UGC examination for lecture, then it is useful for lecturership or assistant professorship in Indian universities and colleges. So this is a very important examination. Then this is DBT BET, that is DBT Biotechnology Eligibility Test, which is conducted annually in two categories, that is category 1 and category 2. And candidates who qualify for category 1 will be eligible for PhD courses with fellowships, and category 2 candidates can join as DBT JRF with fellowship but cannot do their PhDs. Then third is GATE examination, Graduate Aptitude Test in Engineering. Though it is engineering examination, but biology or life sciences students can also appear for this examination, and it is a national level exam which is organized to get admission in IITs, ISERs, IISc, and other reputed institutions in India for MTech, ME, or PhD courses. And it is conducted by IITs or IISc on rotational basis. So, for all of these examinations, you start your studies you may start, I will say, from MSc, and now there is a time to clear those examinations and get admission for PhD. Okay. So apart from these examinations, there are other few examinations, entrance examinations for that respective institute, like NCBS examination, then TIFR, ICMR, JNU, NIPER, then GTU, BITS, Pilani entrance examination, IISc Bangalore entrance PhD examination. So this is the list of exam I have given here. After my lecture, you can go to the link, and if you are interested, you can check the more details about those examinations. So you can see that there are a lot of examinations for doing PhD in India. So for all these things, means for doing PhDs in various government organizations, it needs a lot of funds, and there are different government funding agencies in India. And I think one should know the where should we get fund. So I make this slide. So this is very important funding agency, that is CSIR, that is Council of Scientific and Industrial Research, DRDO, that is Defense Research and Development Organization, DBT, that is Department of Biotechnology, ICAR, that is Indian Council of Agricultural Research, ICMR, that is Indian Council of Medical Research, DST, that is Department of Science and Technology. All these funding agencies provide funds to different government organizations in the form of students' fellowships as well as contingent fund for that for completing their research work. So while doing the PhD, currently the students are getting around 25,000 to 30,000 fellowships from government. Okay, so this is all about fellowships and where and how to join PhD. So now one would think, where should we start or where should we join? Because really we don't know anything in which institute, what type of research is going on, and so on. So here is the top 10 research institutions in India doing research specifically in the field of life sciences. So this is these institutes are IISc Bangalore, then TIFR Mumbai, then Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore, National Institute of Science, Education and Research, that is NISER in Orissa, Indian Institute of Science, Education and Research, ISERS. This is the best place for doing research after your MSc. And there are 7 different ISERS which are located in Bhopal, Pune, Berhampur, Mohli, Kolkata, Tirupati, and Thiruvananthapuram. Then next is NCCS, that is National Center for Cell Science, Pune, Bose Institute, Kolkata, that is West Bengal, then Center for Cellular and Molecular Biology, that is CCMB, Hyderabad, CSR Institute of Genomics and Integrative Biology, IGIB, Delhi, and Indian Agricultural Research Institute, IIR, New Delhi. Okay. So now I would like to explain which are the recent research work is going on in all these reputed institutions throughout India. If we go for IISc Bangalore, the current research is based on modern biology, and the research is going on structural biology, then reproductive and developmental biology, DNA repair, and genomic stability, system biology and bioinformatics, neuroscience in health, disease, enzymology, and so on. So mainly in IISc Bangalore, the research is based on human health as well as various human infectious diseases and drug and molecular design. So you can decide your field depending upon the research work going on in the respective institutions. At TIFR, mainly the research is going on in interdisciplinary manner. That is, there are a lot of departments like biology, chemistry, physics, maths, computer science, and science education. So at the current research work going on is on cell biology, then genetics, biochemistry, and biophysics. So biophysics, this is the field which applies the theories and methods of physics to understand how the biological system works. So this is also a very good field to perform your research in future, and also developmental biology. So these are the various fields where the research is going on in this institute. Then in JNCASR, the School of Biological Sciences brings together cross-disciplinary research and in various areas of molecular biology and genetics and neurosciences. Okay. At NICER, the research is mainly going on host pathogen interactions, cell signaling events, immune cancer microenvironment, molecular events in cancer, then cell biology, and cellular and molecular genetics of aging disorders. So mainly the research is going in the fields of human health, then genetics, molecular biology, and so on. So one can decide their passion or their interest and try to join such renowned institutions for their research work. As I have already told, there are 7 ICERs which aims to shape the nation by inventing and implementing sustainable solutions for societal problems by students through scientific research. So a lot of research projects are going on which are mainly aimed to solve various social problems, and different research work in the field of neurology, cell biology, plant developmental biology is also going on in these institutions. Then NCCS, where currently I am working at NCMR, the research is going on in various fields related to health, such as metabolic disorders, cancer, infectious diseases, and regenerative medicine. The institute is providing valuable services as National Animal Cell Repository as well as National Center for Microbial Resource, that is NCMR, where we supply and characterized microorganisms as a service. And other research areas are stem cell biology, immunology, genomics, proteomics, system biology, structural biology, and microbial ecology. So NCCS has a varied field of research work going inside the institute. Then Bose Institute, here mainly the research is carried out in the field of plant biology, chemistry, molecular medicine, microbiology, and various other environmental science and bioinformatics. And the latest research includes the fields like plant biology, bioinformatics, and systems and synthetic biology. So these are very recent advanced fields where the research is going on in these institutions. Okay, so all of you may know CCMB, that is Center for Cellular and Molecular Biology at Hyderabad, and here the research is mainly carried out as a multidisciplinary areas of modern biology, and the main areas are biomedicine and biotechnology, then genetics, cell biology, bioinformatics, molecular biology, and in the field of infectious diseases. Then important institute is IGIB, where the main research work deals with the development of basic biological research to commercially viable technologies related to healthcare. And here, mainly the work is carried out to understand rare genetic diseases in India. So mainly they focus the human related issues going on in India, as well as they also carried out the research in the field of respiratory disease biology and functional biology and chemical and system biology. And here is IARI, that is Indian Agricultural Research Institute, and it was set up to promote agricultural research. So the students from the field of botany can approach this institution. And here the research is mainly carried out in basic and applied fields. And institute has gained popularity because of the development of commercially available high-yielding major crop varieties and management technologies. So plant-related research is going on in this IARI Institute. Okay. Apart from these very important institutions in India, there are n number of institutes where different research in the various fields is going on. And here I listed the different institutions, that is CSR funded institutions, mainly food research institutes. As our college has started a food microbiology department, so I thought it is worth to show to the students which are various institutions where the food related research is going on. And this is Indian Institute of Toxicology Research, Lucknow. Then IHBT Palampur, where the research on food and nutraceuticals is going on. Then NIIST Thiruvananthapuram, where agro-processing technology and microbial processes and technology research is going on. CFTRI, most of you may know this name. This is the very important and renowned research institution in the field of food science and technology. So this is related to food research institutes in India. Okay, so these are different plant research institutions, that is botany students can really approach these institutions. That is NBRI Lucknow, then IIM Jammu, IHBT Palampur, and CIM AP Lucknow. So you can go to the website and get more information about all those institutions, how to join, which are the examinations, and so on. Okay, so this is also a list of various institutions under CSIR, where a very good quality of research is going on. That is CMCRI Bhavnagar, then NIO Goa, where mainly biological oceanography and marine archaeology research is going on. Then again CSM CRI Bhavnagar, where marine biotechnology and ecology research is they are conducting in this field. NIRI Nagpur is conducting research mainly in the field of environmental biotechnology and genomics, and as well as they are conducting the research in the field of solid and hazardous waste management. NCL Pune is a very important institution in the field of research, where analytical chemistry and material characterization, as well as biochemical and biological sciences related research is going on. IMTech Chandigarh, where the research in the field of exploration of microbial biodiversity, then genetics, molecular biology, biosensors, and nanotechnology. Nanotechnology is also emerging field, and one can really go in depth for research in this field. And then CDRI, that is Central Drug Research Institute, Lucknow, where drug research and development is going on. Okay, so these all are CSR funded institutions in India. The aim of showing all these things to you student is mainly we should approach all those institutes, we should read about them, and you can definitely choose institute of your interest. And these slides, in those slides, I make it compiled list for you so that it will be somewhat easy for you to go through all this vast information. Okay. Then these are the DBT funded institutions, that is National Institute of Immunology, then NCCS, then CDFD. This is a very, very important institution in Hyderabad, and one can really approach this institute if you are interested in fingerprinting diagnostics and such type of work. Then Institute of Life Sciences and National Institute of Biomedical Genomics in West Bengal, and there are n number of institutes under DBT funded institutes are there. Then this is CIAB Mohali, then International Center for Genetic Engineering and Biotechnology, New Delhi, Institute for Stem Cell Science and Regenerative Medicine, Bangalore, and so on. So these all are DBT funded institutions in India. Okay. So this is the list of DST funded, that is Department of Science and Technology funded institutions in India. You all know Agharkar Research Institute at Pune and Bose Institute, then Indian Institute of Astrophysics, Bangalore, and so on. So this is also a big list and you can go through it. Okay. So this is all about what are the different research institutions in India and how you can approach them. There are various examinations. You should definitely go through those examinations, and if you would like to join as a PhD student at those renowned institutions. Now I would like to brief something about work where I am work about our center. That is currently I am working at National Center for Microbial Resource, that is NCMR, NCCS Pune. And this is the single largest collection in the world with more than 180,000 microorganisms. The beauty of the collection is from diverse ecological niches in India, such as soils from Western Ghats, mangroves, marine environment, industrial effluent polluted site, and insect gut. And it played a vital role in exploration and conservation of microbial diversity of India. And these are our mission and objectives, that is to serve as a leading world-class microbial resource repository, provide authentic high-quality services for microbial preservation, characterization, authentication, and supply of authentic cultures to various industries and academic institutions. Also, to serve the nation in various research like biodiversity conservation, biotechnological research, and mainly the research is related to microbial ecology, taxonomy, and systematics, as well as to serve as IDA under Budapest Treaty and DNR under Ministry of Environment and Forests. Okay, so at our main aim of the institute is to provide various services. As I have already discussed, we provide various services such as culture deposition as general deposit, safe deposit, IDA deposit. Then we also provide services in the identification and characterization by using various methods, from traditional method up to genomics and metagenomic methods. Then again, we also provide services in the field of genomics and metagenomics, where we do the genome sequencing of microorganisms, as well as metagenomics was applied to various environmental samples. Then culture supply and the few customized services also we provide at our institution. Okay. So these are the various areas where our institute perform research, mainly in the field of taxonomy, then systematics and biotechnology of microorganisms from various extreme environments using genomics as well as metagenomic approaches. Here we study human microbiome to understand contributions of prokaryotic life to innate working of their eukaryotic host, then investigating biosynthetic potential of microbes for discovery of novel drugs. As already the sir has explained very nicely about novel drug discovery and everything. So this work is going on at our institution. Then understanding the role of microbial communities with concern to the human health. So mostly the research is going on various diseases, that is diabetes and other human health diseases. The work is going on, and studies on specialized group of bacteria like anaerobic bacteria, anoxygenic phototrophic bacteria, and cyanobacteria. So this is very special group of organisms where the taxonomy and biotechnology related research is going on at our institute. And also we are dealing with antimicrobial resistant bacteria. Okay, so after all these information, as per my knowledge, or I think these are various exciting and emerging fields in the field of life sciences, that is neuroscience, then bioinformatics, gene therapy, structural biology, and so on. So under neuroscience, currently what they are doing is they are doing molecular mapping and elucidation of complex brain cell functions. So this is a very important in terms of human health, and it has a direct effect on our society. So this is a very important topic for research for the students. Bioinformatics, as sir has very well explained about, very clearly and very nicely explained about this field. And this is a very important and emerging field for the research, where computer technology is used to solve informational problems in life sciences. So one can definitely go for this field as a part of their research work. Gene therapy, it is based on transfer of genetic material into human for purpose of treating the diseases. This is also a very, very important and emerging field of research in the current scenario. And under structural biology, where you can, it's an interdisciplinary science where you use various techniques from physics, maths, computer, and other sciences to elucidate the structure of a particular part in the human body. So this is also a very important field of research. Then there are a lot of environmental issues like pollution and other environmental related problems. And this is also a very important field of research in the current scenario. Cancer biology, this is a continuously growing branch, and one should do a research in this field of cancer biology and astrobiology. So these are all important and advanced research fields where one can really enter and one can do research and help society in with their solutions. Okay, so I would conclude my talk by saying that recent scientific advancements have led to a brilliant growth in life science industry, and it is not only limited to in-depth research and study, but it has paved the way for a dynamically successful career for individuals who are passionate about the science. So if you are hardworking and if you are passionate and you target your aim, then there is nothing difficult in your life. And I really encourage all of you to enter in this exciting field and really get society, really make society benefited from your research work. So, okay, this is our team at NCMR, and we are working together for last 11 years. And I duly acknowledge DBT Government of India for funding this project, Director NCCS, Dr. Yogesh Choche for his continuous support and encouragement, Dr. Shivangi Kanse Madam from YCIS Satara for her kind invitation and allowing me or giving me chance to discuss this very beautiful topic with all of you and all NCMR staff for their continuous help throughout my tenure. So thank you all, and thank you for patient listening. Thank you.
Okay. Thank you so much, ma'am, for your very informative talk. Now the session is open for the participants. If anyone have a question, then you may put it on in the chat box.
Okay.
Or you can directly ask to the ma'am also.
Yeah, they can ask me directly instead of putting there.
Yes, ma'am. So if anyone have a question, then you can ask to ma'am.
Is there a question? Yeah.
Yes. No, ma'am. Is there nothing question for you? Right.
Okay.
Participants, if anyone have a question, then please you may ask to the ma'am. Okay. So thank you so much, ma'am, for this very informative talk related to the recent trends of research in life sciences. And with this, I request to Professor P.S. Pawar, sir, to deliver the vote of thanks. Over to you, sir.
Thank you, ma'am. Good afternoon to everybody. I am Mr. Pawar B.S. I am here to present the vote of thanks. It gives me immense pleasure to deliver the vote of thanks for the event to dignitaries assembled in online platform. I would like to thank our first resource person, Dr. K.D. Sonone sir, who honored this function with his presence. I would like to thank our second resource person, Dr. Amarja Zoshima, for delivering such nice and informative session. I also would like to thank our beloved principal, Dr. B.T. Jadhav sir, for his support and guidance for organizing such events. And also I would like to thank organizing committee, our vice principal, Professor Dr. V.Y. Deshpande sir, Research Dean, Dr. Mujawar sir, Dr. Kanse ma'am, for organizing such fruitful national webinar. I would like to thank OIT department, especially Doke sir, for his technical support. Thanks to one and all participant, my colleague, for joining or attending such nice webinar. On the behalf of organizing committee, I would like to declare that this webinar is over. Thank you, ma'am.
Thank you, sir.
Okay.